Just for 2 minutes. We'll be waiting for the speaker to enter into the meeting.
Vijayalakshmi ma'am?
Yeah.
Am I audible clearly?
Yes.
Okay.
Yeah. Good morning, ma'am.
Ma'am, good morning. Can you hear me?
Yeah. Very clearly. Nice to have you, ma'am. Today's morning session.
What you are doing?
Myself, Vijayalakshmi from Department of Analysis.
Hello, ma'am.
Actually, because of technical issue, we are little bit late for 2 minutes. Sorry for the inconvenience to the participants.
Can you hear me, ma'am?
Yeah, yeah. Are you able to hear my voice?
1 minute, I'm not.
Yes, ma'am. Are you able to hear my voice?
Yes, ma'am. Yes, ma'am. I'm able to hear your voice now.
Okay. Yeah. Thank you. And now I request Rihanna ma'am to introduce our speaker. Please, over to Rihanna ma'am.
Good morning. Myself Rihanna Sheikh, Department of Pharmaceutical Analysis. I welcome all the participants to second day FDP conducted by Department of Pharmaceutical Analysis at KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. It's my privilege to introduce today's speaker, Dr. Krishnaveni Nagappan. She presently working as a professor and head of the Department of Pharmaceutical Analysis, JSS College of Pharmacy, OT. With 15 years of experience in teaching and research, she was awarded doctorate in pharmacy by the Tamil Nadu Dr. MGR Medical University, Chennai in the year 2012. She has received her MPharm in Quality Assurance in the year 2003 from SNDT Women's University, Mumbai, and Bachelor's in Pharmacy from Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore, in the year 1999. She has published more than 60 papers in national and international peer-reviewed journals. Her research area of interest includes impurity profiling of drugs, pharmaceuticals, particularly in stress degradation studies, characterization of degradation products from pharmaceuticals, and nutrikinetic assessment of functional foods. She has also presented papers in various national and international conferences. She has guided 45 postgraduate students, 1 doctoral candidate, and presently guiding 8 doctoral candidates in the field of stress degradation studies and food analysis. She has received travel grants to attend various national and international conferences and seminar grants from government agencies and JSS AHER Mysore to attend and organize conferences and seminars. Several of her publications has received PD Sethi's Certificate of Merit for the best publication in the field of pharmaceutical analysis. She also received the Best Oral Presentation Award presented at 4th Annual International Conference on Pharma 2016 held at Singapore. She served as Board of Studies member for JSS Academy of Higher Education and Research, Mysore and JNTU Anantapur. She is mentor for DST WOS project worth 18 lakhs. She is also the principal investigator for the DST FIST project sanctioned to the Department of Pharmaceutical Analysis worth 56 lakhs. She had received 2 projects worth 47 lakhs from NCSTC DST and ICMR New Delhi. She had also organized various workshops and conferences in the field of pharmaceutical analysis with fundings from JSS AHER Mysore, DST and ICMR New Delhi. Welcome, madam.
Thank you, madam.
Okay, ma'am. Shall we start the session then?
Yes, ma'am. Yes, we can start the session. Can I share my slides, ma'am?
Yeah, you please share the screen. Thank you, ma'am. We are able to see your screen.
Yes, ma'am.
Come on.
Yes, no, ma'am.
Thank you. And yeah, we are able to view your screen.
Yes, ma'am. Ma'am, how much time I can take? Like 1 hour or 1 hour 15 minutes or something like that?
Minimum 1 hour, ma'am.
Minimum 1 hour, ma'am.
To the max, it depends upon the participants' questions and all.
Okay, fine.
As you know, being
Yes,
an
ma'am,
academician, we know the problem, what we have.
I understand, ma'am. Yes,
And
thank
this
you.
time, PG students, research scholars, they actually participated in this session.
Okay,
Okay, ma'am.
fine, ma'am. Fine. Can I start with the presentation?
Sure, ma'am.
Yeah.
Please.
So to the participants, am I audible?
Yes, you are audible, ma'am.
Yeah, thank you. So a very good morning to one and all. First of all, my sincere thanks for inviting me to give a lecture in this FDP program, which is organized by the Siddhartha College, and my special thanks to Dr. Lakshmi ma'am as well as Dr. Sunita ma'am. Sunita ma'am, we have been, I think, ma'am is there online.
Ma'am, actually, we have exams, university exams.
Okay.
So, ma'am is in rounds and she's going to come back after 10 minutes.
No problem, no
She'll
problem.
join
And
anyway.
yeah, we have been good friends from 2007. Once she came here for attending an FDP program. It was an offline FDP program, QIP program actually. So from that, then we are in touch with. And I sincerely thank both of you for giving me an opportunity to make a presentation in your faculty development
Sincerely,
program.
because of ma'am's suggestion only, and we had that much of profiled, fortunate enough to have you, ma'am. Thank you.
Thank you, ma'am. Thank you so much. Yeah. So today, what I would like to emphasize in today's presentation is the sample preparation technique. Being an analytical chemist, doing our MPharm in pharmaceutical analysis, we learn to operate many instruments. Like we learn to operate UV, we learn to operate HPLC, we learn to operate LCMS if there is an opportunity, or an LCMS instrument, or a GCMS or a GCMS instrument. What plays a major role when we do an analysis is the sample preparation. Because when we want to carry out an analysis, it is not that we always get a neat sample, or it is not that we always get a standard drug. So most of the times we will be analyzing a tablet formulation. When we talk about a tablet formulation, again it is a matrix. It is not only the active ingredient which is present. Along with the active ingredient, there are lot many what we can call excipients which are present. If these excipients are analyzed along with the sample, if these excipients are being analyzed, there may be lot of analytical interferences. Say for example, if we are doing an UV assay, and if the formulation, if the active ingredient has got a lambda max of 270 nanometers, we wanted to perform the percentage purity of the particular formulation. If some excipient which is present in that is also having a lambda max of 270 nanometers, or even if it has got a UV spectral band between 250 to 290 nanometers also, there will be lot of interference which will lead to falsification of the results. Thus, when we are going for analytical method development and validation, extraction of your analyte, which we call it as the sample preparation techniques, becomes a very, very important parameter. So what are the various techniques which are available? Few of the techniques what we have applied here, all those things I will be discussing in this particular presentation. So what is the importance of an analytical study? As I've already told you, we have to, if we get a sample, we need to understand the chemical composition of the sample. We need to understand the physical properties of the sample, and we need to understand the structural or the surface properties of the sample. And why we need to do sample preparation? Just now I have told you, you have to isolate the desired component from the sample matrix so that the other excipients does not interfere with the results. If the matrix is dirty, we need to clean up the matrix. That is, we need to remove the matrix completely. And sometimes what will happen, say for example, if we are going for estimation of heavy metals in water samples and all, especially arsenic, or if you want to estimate the content of mercury and all, the concentration will be very, very less. And we all are aware that each and every analytical technique has got its own quantification and deduction limit. For example, when we are talking about UV, the quantification and the deduction limit may be in terms of micrograms, whereas in case of HPLC, it may be in terms of nanograms. Whereas when we go for sophisticated techniques like LCMS or LCMS, it may be in terms of nanograms, or still it may go down in terms of picograms also. So suppose if we want to analyze some component which is present in a very, very minimal quantity, say in terms of parts per billion, we need to concentrate that analyte so that it is suitable for quantification by the analytical technique what we have selected. Normally, you all will be aware of this. Whenever we are reporting an analytical result, we never report it as not present. We always report it as not deducted by the applied analytical technique. Suppose if I apply a HPLC method for analysis of pesticide residues in food matrix. So if the concentration of the pesticide residue is very, very, let's say in terms of picograms, it may not get deducted by HPLC. So it is like when we report it, we should not say that it is not present. It should be reported as not deducted, and the deduction limit of the analytical technique used is X, Y, Z, whatever we are getting. So that has to be reported. Under such conditions, what we do, instead of taking some 100 grams of the vegetable for carrying out the pesticide residue analysis, we go for taking some 500 grams, or we go for taking 1 kg, and then we go for quantification. So to bring the analyte to the suitable concentration level, also the sample preparation technique is very, very important. So what are the different types of samples what an analytical chemist will normally handle? We may sometimes handle solutions which are ready for injection, say for example, bulk drugs. If we want to perform the assay of bulk drugs, you can directly dissolve it in some solvent, like you can take methanol or acetonitrile, and whatever solvent it is soluble in, you can go for preparing. Sometimes you may get a concentrated solution, which may require dilution or which may require buffering, or you may have to add an internal standard, or you may have to do other volumetric manipulations. Sometimes you may get solid samples, which needs to be first dissolved, like your tablets or your capsules, something like that. So solid samples, which must be first dissolved, and then it needs to be extracted. And sometimes we may go for doing the pre-treatment to remove the interferences and to protect the column or the equipment from damage. So for example, for this is all the biological samples what we go for analysis. Like if you are taking a serum or if you are taking plasma, if we want to do the quantification of the drug, we may be using buffer in HPLC for method development. So when this plasma, if it is not pre-treated, the proteins which is present in the plasma may undergo precipitation when we directly inject onto the column. So to prevent the equipment damage, we go for removing the proteins by certain techniques. Those techniques are now called as a pre-treatment or sample preparation techniques. So your analyte may be found in variety of matrices. We may have biological samples. We may go for analysis of the veterinary drug residues from the meats which are available in the market. Like you may have, if we are talking about chicken, some of the growth hormones which are provided, or the red meat. From them, we go for performing the analysis, veterinary drug residue analysis, or from plant matrices like fruits, vegetables, like you go for a quantification of the functional foods which are present in fruits and vegetables. Say, for example, carrot if we are taking, sweet potatoes we are taking, we talk about the carotenes. Then there are many things, like if we are talking about turmeric powder, we talk about curcumin. So extraction of these functional bioactives which are present in the food matrix. And in food matrix, again, we talk about the contaminants also. So some of the contaminants which are present, like during the processing, there may be heavy metal contamination. When we are growing the particular food compound, you may have contamination, like to prevent the insecticide, to prevent the insects or to prevent the fungal attack onto the plants, we go for using fungicides or we go for using pesticides. So they may stay onto your functional foods what we are consuming. So you have different varieties of food matrices which are available, from which we extract the we extract the required constituent, whatever it is. It can be a, we call it as a constituent. We also call it as a contaminant. Say for example, formulations, when we take impurities are considered contaminants. Cross contamination may take place. Some other drug which is not required is also considered as contaminants. So constituents and contaminants needs to be extracted, and then they have to be analyzed by a suitable analytical technique. So the basic objective of our sample preparation is to free your analyte from other interferences which may give you a false result, and you have to free your analyte from impurities which can damage the column or which can damage the equipment, and you make your analyte compatible with the intended method of analysis. Like solvents, then to dissolve it in the solvent without affecting the sample retention or dissolution, to concentrate the analyte before analysis. So here you can use both the terms. You either concentrate your analyte. If your analyte is over concentrated, you go for dilution of your analyte. Both the things are possible. So when we are talking about quantification, there are four major steps. The very first step is sampling, because when we manufacture lakhs and lakhs of tablets, each and every tablet is not analyzed, it is not quantified for the presence of active phytoconstituent. So what we do, we do the sampling. So how do we do the sampling? Your sample should be a representative of the whole lot of your tablet formulation. Once we do the sampling, the analysis doesn't take place immediately, because the instrument may be very busy. You would have had queues of samples which needs to be analyzed, results to be reported. So the second stage, we call it as the sample preservation. Once you do the sampling, your sample has to be properly preserved. Then your sample has to be prepared so that it is what you can call it can be further quantified, and then we select a particular analytical technique and we perform the analysis. Normally, an analytical chemist doesn't perform the sampling, but your sampling sample normally in industries, the QC chemist goes and he takes the samples. So the sample is obtained from the object which needs to be analyzed, and it should represent the original object, as I have already discussed. Like if it is 1 lakh of tablet, if we are withdrawing some 20 tablets for performing the quality control test, that 20 tablets should be a representative of the whole lot, or it should be a representative of the whole batch of the sample, whole batch of the tablet which is being manufactured. And when you are sampling, you should consider the variability which will occur within the object, and you should also withdraw a quantity of sample which is sufficient enough for carrying out at least 3 analyses. Like you can never perform one quantification and say that the percentage purity is so and so. Even if we are performing a titration, we repeat the titration until we get a concordant value. So the same way, in case of quality control studies, when we are performing a HPLC analysis or when we are performing a UV analysis, we at least perform the analysis minimum 3 times. So you should have required quantity of sample for carrying out the analysis 3 times. And you should also have some control sample. That is, after performing the analysis, some sample has to be stored for further reference. Because once we send the report to the concerned industry, or once we send the report to concerned management, you can call it as CDSCO or somewhere, when we send the result, if there is a contradiction in the result, they may get back to us requesting for further analysis. So under such conditions, we should be able to withdraw the control sample and we should be able to carry out the analysis and prove that whatever we have carried out is correct. May I request Aisha, can you please stop doing the annotations on the slide? It's a kind of disturbance for me. Can you hear me, please? Ms. Aisha?
Okay, ma'am.
Yeah, please. So next is sample preservation. So why we wanted to preserve the sample, as I already told you, immediately after sample withdrawal, we may not be able to perform the analysis because each and every instrument will be loaded with n number of samples. You have to complete one after the other. The first in, first out basis we normally talk as. So we have to follow that. So you have to do the preservation of your sample. So your sample has to be preserved before performing the further analysis. So why we have to preserve our samples? We have to preserve our samples so that there is no change in the physicochemical characteristics of the analyte. Your analyte should not volatilize, your analyte should not diffuse, your analyte should not get absorbed onto the container what we are using for sampling. There should not be any photochemical reactions. There should not be any oxidation of your analyte. Your analyte should not undergo precipitation. Suppose if we are doing a liquid sampling of a liquid, then precipitation may take place. There should not be any precipitation. Suppose if we are taking some biological samples, there should not be any biodegradation or there should not be any enzymatic reaction, which is very, very important for most of the biological sampling what we are doing. And there should not be any change in the sample composition even after collection. So these are some of the parameters what we need to consider when you go for sample preservation. Being an analytical laboratory, sometimes we may be only an what you can call a clinical research organization where we do not do any sampling. So the industry will do the sampling, the industry will send the sample to us, we will be performing the analysis. So this process itself will take some time. After sample collection, they have to do the sample preservation, they will be sending it to us, and we will also be telling like it will take 15 days or it will take 1 month for me to give the result. So that 1 month, how am I going to store the sample? That is also important. That's why we talk about the sample preservation. So we need to go for using proper containers. Temperature control should be there. Sometimes we may have to add some preservatives and we have to maintain the proper sample holding time. So these are some of the examples which are given here. Suppose if you are withdrawing a sample for measurement of pH, the container what you should use should be a borosilicate glass and the pH has to be immediately measured on site. You cannot hold the sample for measurement of the pH. So the pH has to be immediately measured on site. Even temperature also, if you want to measure, you have to go for a borosilicate glass container and the temperature should be immediately measured on site. Suppose if we want to quantify inorganic anions which are present in your given sample, you can collect your sample in plastic or you can collect in glass containers. You can store at 4 degrees Celsius for 7 to 28 days. Likewise, if we are talking about metals, like inorganic metals, like they can be heavy metals, they can be macronutrients, they can be micronutrients. The sample has to be filtered on site. The sample has to be acidified. They can be stored in polytetrafluoroethylene tubes and they can be stored for a minimum of 6 months because the metals may not undergo degradation. If we want to go for estimating organic carbon, hydrocarbons or aromatics or polychlorinated biphenyl substances or something like that, we can collect the sample in plastic or brown glass Teflon tubes. You can hold it between 7 to 28 days and you can store at 4 degrees Celsius. If we are collecting tissues like biological samples, if we are collecting, we have to freeze it till further analysis. -4 to -8 degrees normally we use. Tissues can be held in aluminum foil. If we are collecting a fecal matter can also be held in aluminum foil. And if we are collecting some urine or if we are collecting some blood samples and all, you can immediately convert it into plasma. You can extract the plasma or you can extract the serum on the wherever you collect. At the time of collection of your blood, immediately you convert it into plasma or you convert it into serum. And then you can freeze it between -4 to -8 degrees Celsius in Ria vials or in glass containers until further analysis. So once the sample preparation, how do we do once the sample preservation is done? At the time of analysis, these are the following steps what we carry out. If your sample is a solid matrix, suppose if you have a tissue or if you have a food matrix, the first thing is you do homogenization. First, you defreeze it, that is, you remove it from the freezer. You bring it, we call it as the thawing process. So we bring it back to the room temperature. And once it comes back to the room temperature, it should gradually come back to the room temperature. It is not that you take a frozen sample, you put it in some hot water so that it comes back to the room temperature. No. So you have to just leave it on the table. We call it as working table. Benchtop thawing, we call it as. You just remove the sample from the freezer, allow it to cool down at room temperature, come back to its original condition. Like liquid means it will come back to liquid, or your solid means it can reassign its shape. Then you go for homogenization or you go for size reduction, and then you extract by using a suitable solvent or by using a suitable technique. After extraction, you concentrate it so that you get the required quantity for analysis. Then, if any further cleanup is required, like filtration or before injecting into HPLC or LC-MS, we use syringe filters so that any particulate matter is present after concentration can be completely removed. You do that filtration or cleanup, and then you take it up for further analysis. So these are some of the sample preparation techniques which are being used. Some of the different kinds of sample, what you will be observing is it is a gaseous sample. You may have a gaseous sample, you may have a liquid sample, you may have solid sample. Gaseous samples can be analyzed or gaseous samples can be separated. Suppose if you want to analyze the alcohol content in breath, so you can do liquid trapping, or you can go for purge and trap, or you can go for solid phase micro extraction or stir bar micro extraction technique. If it is a liquid sample, you can go for solvent extraction. Sometimes even you can go for salt sweat extraction. You can go for purge and trap, and all your liquid samples can be handled by any of these techniques. Supercritical fluid extraction you can do, solid phase extraction you can do, liquid phase micro extraction you can do, or you can do solid phase micro extraction technique, or you can do membrane based extraction technique. So any of these techniques we can follow for a liquid sample. Suppose if your sample is a solid sample, you can directly subject it to soft sweat extraction, or you can do go for solvent extraction, or what you can do, you can convert it into a liquid sample. You can solubilize it in some solvent, and then you can go for application of any of these techniques, right from supercritical fluid. You can go for any of these advanced sample preparation techniques. So these are some of the basic sample preparation techniques which are used when we go for analysis of the samples by using various analytical instruments. Suppose if we have organics, most of the hydrocarbons what we are analyzing, you can extract, you can concentrate, you can clean up, that is filtration, or you can go for solid phase extraction, that is removal of the unwanted matter. In certain cases, you may go for derivatization. For example, if your analyte is non-volatile in nature, you have only a GCMS or you have only a GC instrument with you. So you can convert it into a volatile derivative. Then you can use a GC or you can use a GCMS. Your sample is non-volatile in nature and it is not having any chromophore. Then you can introduce a chromophore by doing a derivatization and analyze by using HPLC. So depending on the nature of the analytical technique you're going to select, depending on the nature of your analyte, you can select this derivatization step. It is not compulsory that for all organic samples you will be doing derivatization. You have to just start with an extraction, concentration, cleanup, and if it is compatible with your analytical technique, you can directly proceed with the analytical technique. Either you can go for HPLC or you can go for LCMS. If derivatization is required, you can do the derivatization and you can select the particular analytical technique. If you have volatile organics, you can transfer it into a vapor phase, you can concentrate it and you can analyze by using a GC or a GC-MS technique. If they are metals, you can go for extraction, concentration or speciation. Derivatization, sometimes, for example, iron and all is there. You can extract the iron. You can derivatize it with bipyridyl reagent so that you will get a colored complex. This colored complex can be analyzed by using a UV visible spectrophotometer. So depending on the analytical technique, if you require, you can go for derivatization. Or if you have directly atomic absorption spectrophotometer, or if you have an ICP, inductively coupled plasma, which is again hyphenated with a mass spectrometry, you can just do extraction, concentration, and speciation. Speciation is nothing but if your sodium is existing in the form of a sodium chloride, you have to just convert it into sodium neutral metal. So you do the speciation and then you can go for direct analysis. Suppose if you have ions, ionic substances, this is very common. Polar drugs are there. If your solvent is again a polar solvent, it will easily undergo ionization. Acidic drugs, basic drugs, all these things can easily undergo ionization. Suppose if you have such drugs, you can do the extraction, you can concentrate it. If you want, you can derivatize it, ion pair chromatography. You can use some derivatizing agents, you can derivatize it, and then you can either perform by HPLC, or you can use an ion exchange chromatography, or you can use a UV visible spectrophotometer. DNA, RNA, and all is that means you have to first do the cell lysis, you have to extract it, and then you can quantify it by PCR or electrophoresis techniques or some other techniques. Amino acids, fats, carbohydrates, and all extraction, cleanup. Then you can analyze depending on the nature, GC, HPLC, or electrophoresis you can use. If you have microstructure, then you have to go for etching, polishing, reactive ion techniques, or ion bombardment techniques, and you can measure by surface spectroscopy. So these are so if you observe from this particular slide, if you see the most of your analytes, they undergo extraction, concentration, and cleanup. Derivatization wherever it is required. Suppose if we have a biological sample, because as an M.Pharm pharmaceutical analysis student, after completing my post-graduation or our post-graduation in M.Pharm pharmaceutical analysis, we normally enter into an industrial setup. We work in an industrial setup, whether you work in an R&D, or you get appointed in a clinical research organization, or you get appointed in some bioanalytical or analytical services centers, where they will be having the instrument, they will do the method development, validation, and they will be submitting the results. So some of these, what we call it as CROs, we either join in a CRO, or we join in a QC laboratory, or we join in R&D. Wherever we go, we will always be handling the samples. So when we are handling the samples, your sample may either be a tablet, capsule, or some formulation matrix, or your sample may be a biological matrix. Sample matrix, it is very easy. If it is a tablet or if it is a capsule, you can identify a particular solvent in which your excipients will not get solubilized, only your drug will get solubilized. So you can just extract it with that particular drug and you can perform the analysis. But when we are talking about a biological matrix, lots of issues are involved. And most of the biological matrix, what an analytical chemist or pharmaceutical analysis graduate will handle is blood. Your blood will immediately get converted into plasma, or it will get converted into serum, and it will get stored. Most of the bioavailability studies we do, most of the bioequivalent studies the industry performs. So either for carrying out a bioavailability or for carrying out a bioequivalent studies, either in animals or in human volunteers, maybe a healthy human volunteer, or there you may even have some patient samples to be handled, real-time samples you may have. Suppose you may be doing therapeutic drug monitoring in hospitals. You may withdraw some samples from random patients you may handle. So under all such conditions, we are forced to handle plasmas or serums. In certain cases, especially when we focus on the metabolites, suppose if we have an active metabolite, or if we want to study the metabolic pathway of a particular drug, most of the metabolites are excreted via urine. They undergo the first phase metabolism, they get excreted via urine. So in certain cases, to do a metabolic profiling of a particular drug, we use the urine samples. Fecal samples in certain cases are used. Suppose if the drug is poorly absorbed, or if the drug is extensively excreted, or it gets converted into a metabolite, we handle the fecal samples. Salivary samples are also handled in certain cases, especially we all are aware of this microbius ring, which gets dissolved in the saliva and then it enters into the bloodstream. So under such conditions, saliva, breath. Suppose if toxicity studies needs to be performed, or if you want to understand the deposition of the drug in various tissues, especially pharmacology experiments they will be doing, animal experiments they will be doing. So how much drug gets deposited on a particular tissue, how much drug gets targeted onto a cancer cell. So for all such studies, we go for using this tissue samples for analysis. So we go for handling, depending on the nature of the study, depending on the nature of the analyte, we go for selecting any of these biological samples for further analysis. So these biological samples have first, they should undergo preliminary treatment before you convert it into your matrix, which needs further cleanup. This we call it as the preliminary cleanup, or we call it as the preliminary treatment. Normally what we do is we do a very simple protein precipitation or denaturation. You go for adding some salts like tungstic acid or ammonium sulfate, or you slightly heat your biological sample, it automatically the protein will get precipitated, or you add some organic solvents like alcohol, or you add some acids like trichloroacetic acid or perchloric acid, or some organic solvents like methanol or acetonitrile, automatically the proteins will undergo complete precipitation. The supernatant can be withdrawn. This supernatant can further undergo certain cleanup processes, and then they can be used for further analysis. Um, if the proteins are high molecular weight proteins, you can just go for ultrafiltration, or you can go for dialysis. Depending on the pore size of the membrane, these proteins will get retained. And most of our pharmaceuticals, what we are analyzing, are all small molecules. So these small molecules may cross your ultrafiltration barrier or your dialysis barrier, so that that can be further collected, concentrated, cleaned up, and analyzed. You can go for hydrolysis also. Suppose if the drug is in the form of a conjugate, if you want to convert it into the actual component, you do either enzymatic or you go for acid hydrolysis. We go for homogenization, especially in case of tissues. Any tissues what we are taking, we have to first homogenize, and then we have to go for further analysis. So this is the preliminary treatment. The very first thing what we are seeing here, that is the protein precipitation or denaturation process, is itself a sample preparation technique. And I cannot say that there may not be any interferences, but denaturation or your protein precipitation is itself sufficient. Suppose if we take plasma samples, you add these precipitating agents, the protein will get precipitated, the drug will be there in the supernatant, which can be filtered and which can be used directly for further analysis. What problem we will face here is only if your drug is having a very high protein binding. Your drug binds 80% to the proteins, then it becomes very difficult. So under such conditions, you cannot go for protein, because when your protein is precipitating, along with that, the drug which is bound to the protein will also undergo precipitation. So these are some of the extraction procedures which are used the most. As I've already told you, protein precipitation, the cheapest and the most simple extraction technique or a sample preparation technique, we call it as protein precipitation. We have, then we have liquid extraction technique, we have solid phase extraction technique. Under solid phase extraction, we have the solid phase micro extraction and Stirbar-Sopkin extraction technique. So protein precipitation, as I've already told you, it is very useful for messy, dirty protein samples, and it's used in bioanalysis, suppose plasma or serum, especially for removal of proteins. You can denaturate your proteins by using some acids, bases, or some salts, or by using some organic solvents. This is not an exact extraction method. This is just a sample preparation, a basic sample preparation, or you can call it as a basic sample cleanup procedure. It has got its own disadvantages, which is not selective. The extract itself will be dirty, it will not be very clear, it will be colored, and all those things will be there. Proteins, peptides with less than 5,000 Daltons may not get precipitated. Additional cleanup like filtration is required. It is not a highly sensitive technique, because your sample will get diluted. You are adding 1 ml of methanol, or you are adding some 1 ml of 0.1 molar trifluoroacetic acid or something like that. So your sample will get diluted, and you will have a very high background noise when you do only the protein precipitation technique. So this is just an example of a chromatogram. We have estimated the amount of testidecaprom oxalate which is present in rat plasma by using a protein precipitation technique. So you would be able to see here, this is one acetylopram oxalate, and this is the, this is the internal standard, and this is the acetylopram oxalate. And you can see here, this is the interference which had arisen from the dirty plasma matrix, what we have subjected for protein precipitation technique. So we normally think protein precipitation, we are extracting the analyte. Actually, we are not completely extracting the analyte. Along with the analyte and the internal standard added, you will also have some matrix impurities which will also get extracted from the sample. So this is because of that. So normally when we are developing a HPLC method, we see to it that your analyte gets eluted after 5 minutes. When we are using biological samples like plasma or serum, when we are using, we normally focus, first we develop the method. No? So when we are developing the analytical method only, we keep in mind that the drug should elute only after 5 minutes, because most of these polar impurities will elute immediately after the dead volume or immediately after the dead peak. So all those things will get eluted before 5 minutes, and after that the analyte gets eluted. So the second technique is a liquid-liquid extraction technique, the most commonly used technique. Here the separation is based on partitioning between the organic phase and the aqueous phase. We will be using two phases. So the drug will undergo partitioning between the organic, a very simple partitioning, what we normally do in laboratories. So when we are studying our B Pharm, we would have done an experiment on iodine, carbon tetrachloride and water, how much gets partitioned. The same principle is used here. So if we have a hydrophilic analyte, the hydrophilic analyte will prefer the polar phase. If we have a hydrophobic compound, the hydrophobic compound will prefer a organic phase. So here one phase will be aqueous, the other phase will be an organic solvent. And depending on the solubility of your analyte, the analyte will get separated. But before considering the liquid-liquid extraction technique, we need to consider the following parameters. Like here, the first thing is the solvent. What solvents you are going to select? We are going to select two solvents. So what are those two solvents? The two solvents should not be miscible with each other. First criteria, whatever solvent, two solvents we are selecting, the two solvents should be non-miscible with each other. But the drug, whatever we are going to extract, or we call it as the components, the components should be miscible in the solvents what we are selecting. Second one is the density. The solvent which are more denser than water, they will form the lower layer of the pair when mixed together, while solvents that are less dense than water will form the upper layer, or they will float on top of the aqueous phase what we are going to use. Say for example, plasma or serum we are going to use. So you have to select the solvent. So denser solvent will form the lower layer. The drug will get the drug will permeate into your organic solvent, and you can just open the separating funnel. You will collect the organic solvent. Otherwise, decantation and all those processes will be there. So select density should be important, and the solubility is also very important. So both the solvents should form visibly distinct phases when mixed together, and even if they are somewhat visible also, if you allow it to stand for some time, they should get completely separated. So recovery should also be there. This is very, very important when we are handling our analytical analytes, our biological matrices. We normally talk about a recovery between 80 to 100%, because complete extraction is not possible. If we all remember, when we are doing bioanalytical method validation, there is one parameter called as accuracy. In accuracy, we check the recovery of the analyte from the biological matrix. So when we are doing this bioanalytical method validation, what we do is we spike the drug into the plasma, and then we try to check how much of the drug can be completely extracted from the plasma. And that sample, the result what we get for that sample, we compare it with the neat analyte, we call it as, that is a standard 10 microgram per ml solution or 100 microgram per ml solution, and we see how much can be recovered. Any recovery greater than 80% is acceptable. So if we are using some aqueous, or if we are using some organic solvents, The analyte should distribute themselves between the aqueous as well as the organic layer according to the Nernst distribution law, and they should have relative solubility in both the solvent, and the effect of distribution on the extraction causes, the percentage recovery, the nature of the analyte, and the effect of concentration on the recovery all should be considered. Normally, when we are using this liquid-liquid extraction, it is not one-time extraction. It should be so the extraction, we have to repeat the extraction. First, you add 1 ml of organic solvent, you remove it and keep. Then again, because it is partitioning, depending on the partition coefficient, we have to repeat the extraction steps. So the quantity of the analyte which is being extracted in your organic solvent depends on the distribution coefficient, depends on the volume of the two phases which are used, because we all are aware of forming a saturated solution. So ratio of the volume of the two phases used, then utilization of the multiple portions of the extracting solvents than using single portion, that is what precipitation. And yeah, the recovery is also independent of the concentration of the original aqueous phase. Here, it's a very simple, I don't want to explain everything in detail. We normally do it with a separating funnel. If we are handling very, very small quantity of the sample, we normally do it in a Rhea vial or we do it in an Effin drops tube. So you put your organic and aqueous phase together in the separating funnel, you shake it for some time, you allow both the phases to get separated, and then you remove the required phase. With either through the top or through the bottom of your separating funnel, you remove the required phase. So while separation, you can go for using some cotton adsorbent so that if there is some emulsion formation can be prevented. So again, you repeat the process. In your aqueous phase, again, you add some quantity of organic phase and you repeat the process until you assume, because we have to assume here, we cannot tell that, yes, my whole drug gets extracted. So until you assume that whole of your analyte would have got extracted into your organic phase. Apart from this, we also have this continuous LLE, continuous liquid-liquid extraction, which is a kind of your soft set extraction process. So it is applicable when the distribution coefficient of the analyte between phase A and phase B is very, very low. Suppose if the distribution coefficient is very, very low, you can go for using this particular technique. So this is a multiple extraction procedure, like use of your soft plate apparatus. The lower phase will get accumulated here in your round bottom flask. You will be heating your round bottom flask so that again it evaporates, it condenses, only your solvent will evaporate, it will condense, it will again come into contact with the sample. Then the sample will get, that is the analyte from the sample will get extracted and it will get stored in the round bottom flask. So advantage of this technique is it will avoid emulsion because we are not doing any shaking. And this is like we perform on the principle that the organic solvent cycles continuously through the aqueous phase because of constant vaporization and condensation of the extractive agent. So it is designed for high density solvents when compared to that of water. And this is just the explanation of it's just an example I have given here, which I have got from some of the standard books. Like suppose if I have a sample, if I have a matrix which is containing a mixture of organic compounds, say for example, I have a sample, a matrix which is containing a base, which is containing a weak acid, which is containing a strong acid, which is also containing certain neutral analytes, then we have to go for fractionation method in liquid-liquid extraction. So most of the pharmaceuticals, most of the pesticides, what we are going to analyze, they all may be ionogenic or they may be neutral. We all are aware of this technique. That is where our ion pair chromatography or ion exchange chromatography is playing a major role. The drug may be acidic, the drug may be basic, the drug may be neutral, all those things happens. So such drugs, suppose if you have a mixture of drugs, one is acidic, one is basic, one is strongly acidic, one is weakly acidic, one is a neutral, you can go for using this particular recovery procedure, which we call it as a fractionation procedure. And this procedure is based on the pH control of the aqueous sample. You control the pH of your sample so that the drugs can be extracted one after the other. So this is a very, very simple example. Now our sample is containing inorganics, our sample is containing a basic drug, our sample is containing strong acids, weak acids, and neutrals. So what we first do is we convert the pH of our sample to pH 2. The moment we convert it into pH 2, the inorganics and bases will stay in the acidic phase. That is, in your aqueous phase, the inorganics and the bases will stay. To this, you add an organic phase. So, strong acids will not undergo ionization at pH 2. Weak acids will not undergo ionization at pH 2. Neutrals will not undergo ionization at pH 2. So, all the three, the strong acids, weak acids, and neutrals will get extracted onto the organic phase. So the aqueous phase will only contain the inorganics and the bases, depending on their distribution coefficient value. Now what we do, we take this solution, your sample, which is at pH 2. Now you convert it to pH 10. That is, you adjust the acidic pH 2 to pH 10. And again, you extract with an organic phase. Now your base will become non-ionized form, because previously it was in an ionized form. The moment it goes to a basic pH, pH 10, it will become non-ionized. Inorganics will of course be soluble in water. So they will stay in the aqueous phase, and your bases can be extracted in the organic phase. So a basic drug is now completely extracted in the organic phase. And the aqueous phase is now containing only the inorganics. Now you take the organic phase, which we have previously seen here. Yeah, here. This organic phase is containing strong acids, weak acids, and neutrals. So we take this organic phase and we add a bicarbonate solution so that the pH becomes 8.5. So at 8.5, strong acids will get retained in the basic phase, whereas weak acids and neutrals will move to the organic phase. So this organic phase, we are adding a bicarbonate solution. It's an aqueous solution. So only strong acids will get retained in the aqueous basic phase, and the weak acids and the neutrals will move towards the organic phase. Again, this organic phase, we add sodium hydroxide, that is pH 10. So weak acids will undergo complete ionization because it is a highly basic pH. So weak acids will undergo complete ionization. They will get retained in the aqueous phase, and neutrals will get retained in the organic phase. So now we have separated the neutrals in the organic phase, and we have an aqueous phase where weak acids are present. We also have an aqueous phase where strong acids are present, and we also have an aqueous phase where inorganics are present. So we have the three aqueous phases. So these three aqueous phases are now converted into, that is your strong, that is this aqueous phase which is containing the strong acid, you again convert it into pH 2. So that what will happen? They become non-ionized, they get extracted in the organic phase. The same way, the aqueous phase which is containing weak acids also, you convert it into pH 2. Again, they will become non-ionized, they will get extracted in the organic phase. And your inorganics, you have to analyze only in the aqueous phase. So likewise, you are playing with the pH. When you have different types, that's where the pKa value plays a major role. The pKa value is very, very important for analytical method development. In addition, the pKa value is also important for sample extraction. Suppose if you add a very strong acid and your analyte is a base, it will completely undergo ionization. You can never extract it with the organic phase. So we have to be very, very careful. The pH plays a major role. So adjusting the pH, you extract the sample one after the other into the organic solvent, and then you go for further analysis. So these are some of the characteristics of liquid-liquid solvent extraction solvents. Your solvent should have low solubility in water. It should be volatile for easy removal and concentration. It should be compatible with the diffusion technique. I cannot use benzene, which has got a lambda max of 250, 254 nanometers. So it should be compatible with the diffusion technique. Even if I use benzene or if I use toluene, I have to completely evaporate it. And the polarity and the hydrogen bonding properties should also be there, so that the analyte recovery will be very high and the solvent should be of very high purity so that it will not contaminate the sample. So some of the solvents which are used, pure water is used, acidic solutions, basic solutions are used, high salts, complexing agents are used, a combination of above are used as aqueous solvents. Water immiscible organic solvents, we have aliphatic hydrocarbons, diethyl are the most commonly used, dichloromethane, methylene chloride, chloroform, ethyl acetate, aliphatic ketones, aliphatic alcohols, toluene, xylene are used. Water miscible organic solvents, we use alcohols, ketones, aldehyde, carboxylic acids, acetonitrile or dimethyl sulfoxide. There are lots of problems associated with this LLE technique. The most important problem is emulsion formation because we use a separating funnel, we shake it. So emulsion formation will be there. Sometimes analyte may get strongly absorbed onto the particulate matter which is present and they may get bound to high molecular weight compounds. Some compounds will have 50-50 solubility in both the phases. So you need to repeatedly extract. So how can you overcome these problems? Emulsion formation can be overcome by adding a small quantity of salt to the aqueous phase. You can heat or you can cool the extraction vessel. You can filter it through the glass bowl or you can filter through phase separation filter paper. You can add a small amount of different organic solvent or you can go for centrifugation. Absorption, suppose if particulate matter is formed, continuous washing of the particulate matter with a stronger solvent will help you to relieve from this problem. And this is one example of a drug, oxcarbazepine and lecarbazepine. Lecarbazepine is a metabolite of our oxcarbazepine. We have used a liquid extraction technique, and then the final sample which is being analyzed by using a ultra performance liquid chromatography, MSMS, tandem mass spectrometry technique. So these are the TIC chromatograms, total ion chromatograms of oxcarbazepine. Oxcarbazepine D4, which is used as an internal standard because nowadays most of the regulatory agencies are talking about deteriorated internal. This is actually a work which was carried out in industry. So oxcarbazepine D4, which is used as a deteriorated standard, which is used as an internal standard. And this is 10 monohydroxyoxcarbazepine, which is nothing but the lecarbazepine, which is the active metabolite of oxcarbazepine, and its deteriorated internal standard. So what we have done here, since it is an LC-MS-MS technique, we have just taken 100 microliter of the blank plasma. We have spiked it with 50 microliter of the total drug solution. That is, the drug solution will contain oxcarbazepine standard, deteriorated oxcarbazepine, lecarbazepine, deteriorated lecarbazepine. So we have spiked it with 50 microliter of the standard drug solution. Then we have added 10 microliters of 10 millimolar ammonium formate solution. So it is a process of acidification of your plasma sample. Then this is the extracting solvent what we have used. Your extracting solvent, this is the aqueous phase, this is the organic phase. Your organic phase may not be a single solvent. It is not that you use only toluene or you use only diethyl ether. You can even go for combination because this is trial and error. You do many trial and error technique and you fix up with one combination which will give you the highest recovery. So here in this case, we have used 2.5 ml of diethyl ether, dichloromethane in the ratio of 80:20. The sample was vortexed for 5 minutes and then it was infused at 4000 RPM for 5 minutes at 4 degrees Celsius. Then we have removed the supernatant. The supernatant was evaporated to dryness under nitrogen atmosphere. We cannot heat again. If we are heating, the drug may undergo degradation. So it was evaporated under nitrogen atmosphere. Then it was reconstituted with the mobile phase. And it was analyzed by using UPLC MS-MS. And we have to be very careful here because sample loss will be there if we are continuously changing the container. So from here, we are completely removing the supernatant. In the same container, you dry it and then you reconstitute it. So reconstituted with the mobile phase and it is analyzed by using a LC MS-MS technique. And these are the TIC chromatograms. And these are the blank chromatograms. Yeah, so at the retention time of the drugs, we are not having, sorry, these are the LOD chromatograms of the drugs and the deteriorated standards. So if you have binding to proteins, if the drug is getting bound to protein, you can go for adding a detergent, you can go for adding an organic solvent or a strong acid, and we can go for dilution with water, or we can go for displacing the protein binding capacity with a more strongly binding compound. This LLE has got its own disadvantages. You need a large quantity of solvent. It is a labor-intensive process. One after the other, the sample has to be analyzed. It requires evaporation step. More than one extraction is required, as we have already discussed. Suppose if you have mixture of analytes, you have to do 4-5 steps so that you extract all your analytes into the organic phase. Emulsion and contamination issues are also possible in case of liquid-liquid extraction. So this is one advanced technique of liquid-liquid extraction, which we call it as the micro extraction on a glass chip. So this is the micro extraction unit. You can see here micro extraction channel, which is fabricated on a glass chip. So these are the small tubings, micro extraction tubings. This is the micro syringe pump what we are using. One channel will introduce an organic phase. One channel will introduce the aqueous phase. So this aqueous phase will be containing the analyte. So when they are flowing through this microchip channel, both the organic and the aqueous phase will come in contact with each other. The analyte from the aqueous phase will get diffused into your organic phase, and then they move out to the drain. So anything which is coming out of your organic phase is collected and it is further used for analysis. So the advantage is like your extraction will take place at a faster rate, especially when the concentration of your sample is very, very less in terms of microgram quantities. If you have, the extraction will take place within 45 seconds, and it requires no mechanical stirring, mixing or shaking, so that the emulsion formation and all those things can be completely avoided. And this is one more technique where your solid support, you have a solid support, and then you have the two two phases, liquid-liquid extraction. So we call it as a solid supported liquid-liquid extraction technique. This is the solid support which is used, and it is a 96 channel programmable robot liquid handling workstation. So first you will be in that first you will be having the solid support coating. Onto that you will be adding the aqueous phase, and then you will be adding the organic phase. So the separation will take place between the aqueous and the organic phase. Sometimes the sample also will get absorbed. That also will be extracted by using the organic solvent, and then it will be further used for analysis. And the most sophisticated technique what we are using nowadays, and it is a little expensive technique also, is called the solid phase extraction technique. We go for using, it is like a mini chromatography technique what we are going to perform. We go for using chromatographic particles, packed bed column cartridges or similar cartridges are being used. It's a well-established commercial technique. Lots of literatures are available. This is a process by which you will get a very, very clean extract. No additional constituents will be present. Recovery will be very, very good for polar analytes, but your sample must be in a liquid state. Here, gravity, pressure, and vacuum plays a major role, and it is an automated process. So at a time, 10, 15 samples can be processed. Suppose if we are doing bioequivalence studies, and if the number of volunteers, if the study is being conducted in 24 volunteers, you can just imagine per volunteer 13 samples. Time periods, we have minimum 13 samples. So 13 into 24, and a bioequivalence study is performed in 2 phases. So 13 into 24 into plus 13 into 24. So those number of samples needs to be separated. So automation will help us a lot. So here again, it is again the same. Distribution coefficient plays a major role. You pass the liquid sample through your adsorbent. Your adsorbent will absorb the analyte. You wash the unwanted matter, and then you use a liquid which will dissolve your analyte from the adsorbent and gets extracted. So the types of the SPEs, we have the solid phase extraction, solid phase micro extraction, stir-burst octave extraction. It is a non-equilibrium or it is a pseudo-equilibrium procedure. And the basic process, as I've already told you, there are 2 things. You either have adsorption, you also have absorption. So adsorption is a 3-dimensional matrix. So it is like the uptake of water by a sponge. The analyte will the first one is absorption. So it is the attraction of an analyte to a solid that results in accumulation of the analyte's concentration at the porous surface of the solid. Adsorption is again a two-dimensional process, and it results from weaker interactive forces than absorption. So either your process is adsorption or absorption. You cannot easily distinguish whether your drug is undergoing adsorption or absorption. So we normally call it as sorption. Your drug gets sorbed onto the what you can call the extracting medium. That's we simply refer to as a sorption process. And the stationary phase what we use is called as a sorbent. It is a solid extracting phase. It contains solid supported liquid phases also. In certain cases it also contains solid supported liquid phases. And now we have to understand what kind of analyte you are going to take and what kind of sorbent we are going to select. N number of sorbents are available. So what are the different mechanisms by which your analyte may interact with the solid sorbent? There are basically 4 ways. As we have already said, absorption is one way. They may interact with the sorbent by just penetrating into the 3-dimensional structure of your sorbent, or they may undergo hydrogen bonding, van der Waals dipole-dipole interaction. Sometimes there may be surface interactions. There may be intermolecular forces which are involved. So there are a lot other mechanisms which are involved. Suppose if you use a porous sorbent, there can be diffusion. We call it as film diffusion, pore diffusion, or adsorptive reaction. So film diffusion, that is your analyte passes through a surface film to the solid phase surface. Pore diffusion, the analyte passes through the pores of the solid phase. Adsorptive reaction, the analyte may bind or associate or interact with the sorbent surface. Again, if your compound is ionizable in nature and if your compound is in aqueous solution, as we have discussed previously, you can go for selecting certain sorbents which can have ionic interactions, like ion exchange sorbents can be identified. So depending on the nature of the analyte, as I've already told you, you have to go for identifying the sorbent. There are 2 major requirements for a successful solid phase extraction to take place. The technique should be highly reproducible, and your analyte should be taken up by the sorbent, and the process should be reversible. That is also important. The sorbent process, the sorption process should be reversible. So your sorbents must be porous with large surface areas. They should be free of leachable impurities. They should have high stability towards the sample matrix, and they should have high stability towards the elution solvents which we are using. So some of the solvents what we use, there are many solvents which are being used. Suppose if you are using a normal phase mechanism, if your compound is highly non-polar in nature and you want to go for using a normal phase Spme cartridge, silica, alumina, magnesium silicate, or some of the highly non-polar reagents which are available, polar stationary phases which are available. In addition to this, modified polar stationary phases are also available, like aminopropyl stationary phases. We call it as the mid-polar stationary phases. So silica bound to, that is, we call it as the bonded stationary phases. We all would have studied when we were talking about the C18 in, I think in first term form, second semester syllabus, it is there, bonded stationary phases. So these are some of the bonded stationary phases, like mid-polar bonded stationary phases. Like aminopropyl bonded stationary phase, or cyanopropyl stationary phases, or diol stationary phases. These are some of the modified bonded stationary phases which are used, which have got a mid polarity. These are some of the examples of the polar stationary phases what we are going to use. They are mainly used to remove the matrix interferences from organic extracts of plants and animal tissues. And the hydrophilic matrix components will be retained, whereas the non-polar compound will get eluted out. So the interaction between the hydrophilic matrix and your sorbent may be due to hydrogen bonding or dipole-dipole interactions, or pi-pi interactions, or induced dipole-dipole interactions. Then we have the apolar polymeric resins. We are aware of all these things. Converting your silanol into C18, or you're converting it into ion exchange resins, we can go for doing this. They are called as bonded stationary phases. And these are the bonded silica sorbents which are available, C18 sorbents which are available. These bonded stationary phases, normally C18 cartridges are available. So these C18 cartridges are non-polar cartridges. They are very good for separation of polar or ionic substances which are present. They can be used in a pH range between 2 to 8, like how we use our HPLC column. The same way you can go for using between 2 to 8. And graphitized carbon sorbents are also nowadays used. There are two types of graphitized carbon sorbents. One is graphitized carbon black and one is porous graphitic carbon. And they are mainly used for polar, extremely water soluble organic compounds from aqueous samples, because aqueous sample extremely water soluble, they cannot be partitioned at all. So under such conditions, we go for using these graphitized carbon blacks or porous graphitic carbons. Then we also have functionalized polymeric resins, like macroporous polymers, which are containing two monomers, like divinyl benzene, which is lipophilic in nature, and the N-vinyl pyrrolidine, which is hydrophilic in nature. They can help you to separate both by two mechanisms, that is hydrophilic, lipophilic mechanisms. They can balance and they help you to separate. So functionalized polymeric resins are also nowadays available, depending on the nature of the analyte. We also have the ion exchange sorbents. Again, ion exchange sorbents, again, strong cation exchange resins, strong anion exchange resins, weak cation exchange, weak anion exchange resins are there, which are mainly used for irreversible ionic analytes. Suppose if you are analyte, previously we have seen an organic, strong acid can be converted into ionized form and non-ionized form by changing the pH. Suppose if it is an irreversible ionization mechanism, you can go for using these ion exchange solvents. We also have controlled access solvents. So these controlled access solvents, they are mainly intended to be either inclusive or exclusive. They are mainly used for macromolecules, large molecules, or they are mainly used for macromolecules. This procedure is like a gel filtration chromatography technique. That is, you will have a particular pore size of your sorbent. Only one particular analyte can pass through. That is, if your protein is of larger size than your analyte, they will get eluted first. Your analyte will pass through all the pores, they will get hindered, and then they will get eluted. So it will allow accessibility of only macromolecules to the internal pore structure of the solvent so that they get retained. So this is an example. We call this as restricted access media. They are all called as controlled access solvents. They are also called as restricted access media. They will try to retain all the small molecules, because all these small molecules can pass through these pores and they get retained, whereas the large molecules like the unwanted macromolecules like the proteins and all will easily pass out. So this is a kind of what you can call sieving technique, molecular sieving mechanism, what we have discussed. You would have learned in chromatography also, gel filtration, gel permeation chromatography, same mechanism is used here. You also have molecularly imprinted polymeric sorbents. These are some of the latest. We call them as MIPs. They are very selective for specific. This is like an antigen antibody reaction kind of thing, lock and key mechanism kind of thing. So one particular molecularly imprinted polymeric sorbent is specific for only one particular analyte. That analyte only will get held, whereas the remaining analyte will get removed off. So such sorbents are also available. They are called as molecularly imprinted. That is very specific for a specific kind of analyte. And mixed sorbents are also available nowadays. Why mixed sorbents? Because your sample may have a polar analyte, it may have a non-polar analyte, it may have some large molecules also to be analyzed. So mixed sorbents are also nowadays available for performing the solid phase extraction technique. Okay, so we have seen these many things. There are different types of sorbents which are available, like different types of HPLC columns available. In the market, there are different types of sorbents available. So how are we going to select? Because all these sorbents are costly. A very, very small cartridge we will be purchasing and we will be performing the analysis. So I cannot purchase different types of sorbents. I cannot keep it in my lab and perform the analysis. So how do we select what is required, which sorbent I am going to use? So for that selection, we need to understand the following points. That is, first thing you have to check. Is your sample matrix miscible with water or organic solvents? If your sample matrix is water soluble, then you have to find out the next point. Is the analyte ionized or non-ionized? If your analyte is ionized, Is it permanently ionized? That is pH independent. Any pH you put, it will be in the ionic form. So if it is permanently ionized, it is better you go for ion exchange cartridge. But if it is pH dependent ionization, then you can go for anionic. You can check whether it is anionic or cationic. Accordingly, you can select, or you can modify your pH so that you make it into non-polar, and then you can use the C18 column cartridge. And if the sample is organic solvent miscible, is it mixable only in non-polar organic solvents like hexane or toluene, or is it miscible with polar organic solvents like acetonitrile or methanol? That also we have to study. This will give you an idea whether your analyte is non-polar or polar or moderately polar in nature, and accordingly go for selecting the sorbent. So this is the selection guide for the sorbent sample. Of course, this is like your HPLC only. I don't want to explain much on this particular slide. Depending on the nature of your sample, how do you select our HPLC columns or an LC-MS columns? The same way we go for selecting the. So you can just imagine it. This is a miniature of our HPLC. The only thing we are not using any pump. That's it. So these are some of the primary interaction mechanisms like van der Waals, polar dipole-dipole interactions, hydrogen bondings, and electrostatic interactions which will take place. And this is how the cartridge will look like. The cartridge will have a lever tip, then it will have a frit, because this material, the sorbent material, should not enter into your sample. So you have the fritted disc which will not allow the particulate matter to pass through. Then you have a sorbent bed which is 1 centimeter in height, and the particle size is around 40 micrometers. Again, you have a fritted disc which is made up of polypropylene, and this is the sample reservoir. And when we are doing this SPE, we have 5 steps. One is conditioning, loading, washing, elution, and then we have the analysis. So what do we do with conditioning? Conditioning, you wet this. That is like we say equilibrating the column in HPLC. The same way we condition the cartridge with organic solvent as well as we condition it with aqueous solvent. You pour 1 ml of organic solvent, you condition it. You pour 1 ml of aqueous solvent, you condition it. Just you put it into the waste. Then you add your sample matrix. Sample matrix is containing your analyte as well as the matrix. You allow your sample matrix. You have to apply vacuum here because the particle size is very, very small. Gravitational force will not help us. So you apply the vacuum. So your sample should be a liquid sample, which is containing the analyte. You pass through, when you apply the vacuum, the liquid will elute out. The analyte will get sorbed onto the sorbent what you have selected. After that, what we do, we wash it normally with water. So we wash it with water so that any what you can call any impurity or any contaminant which is soluble in water will get eluted out. So all these green particles are nothing but a contaminant. So they will get eluted out, and then you go for eluting. For elution, you either use the mobile phase, what we are going to use for analysis, or you go for using the organic solvents. Depending on the nature of the analyte, you go for using the organic solvents. You elute it and then you further analyze. So some of the solvents which are used like HPLC only. How do we select the solvent? You can select one single solvent, or you can select a mixture of solvents. Sometimes you can select acidic solvents, like previously we have seen 10 millimolar ammonium acetate, a mixture of acidic aqueous as well as organic phase. Anything can be selected. So n number of opportunities are available for selection of the elution solvent. But previously for conditioning, for washing and all, we normally use water and organic. One single organic solvent we go for using. But elution solvent, n number of opportunities depending on the nature of the analyte. And these are some of the advantages. More complete extraction of the analyte is possible. Various SPE formats are available. It is more efficient separation of the interferences. Then reduced organic solvent consumption. We don't use much like previously. Repeated extraction and all we do not do here. One single extraction. So reduced organic solvent consumption. Easy collection of the analyte fraction, and it is more convenient. It can be automated. So it's much easier, less time consuming. Very, very important. And this is one example. I think I have to just. Yeah, so this is one example. I think this is pantoprazole. Yeah, pantoprazole and levosulfuride, simultaneous quantification of pantoprazole and levosulfuride. And we have used iloperidone as the internal standard. We have followed this SPE extraction procedure. Like we have taken 1.7 ml of blank plasma and we have spiked it with 300 microliter of the standard drug solutions. We have vortexed it for 5 minutes. Then we have extracted it using this C18 1 centimeter strata cartridges. So for conditioning, we used acetonitrile and water. Then we have loaded the sample. We have washed it with millicube water, and then we have eluted it with 1 ml acetonitrile, because both the drugs what we have taken and the internal standard, they were all freely soluble in acetonitrile. So we have just extracted it with 1 ml of acetonitrile. The acetonitrile was evaporated to dryness, and it was reconstituted with the mobile phase, and then it was analyzed by using the LCMS. So these are the blank chromatograms, and these are the TIC chromatograms of the drugs. So this is for pantoprazole, this is for levosulfuride, as well as this is for iloperidone, the internal standard. Ma'am, can you hear me? Yeah. So this is just a comparison between the LLE and the SPE in order to understand the advantages. In case of LLE, we use a large quantity of solvent. The maximum in SPE is 20 ml. This is a continuous process. This is just a filtration process. Here, emulsion formation is there. There is no emulsion formation. The selectivity is very, very limited here, depending on the nature of your analyte. But here we have wide selectivity of solvents, and it takes a long time for sample separation, whereas here the time taken is very, very minimal. And these are some of the advantages of the process. So few of the labor, yeah, it is a labor intensive method development. Sometimes some of the disadvantages, again, each and every technique will have their own advantages and disadvantages. So it is a labor intensive method development. Evaporation steps are often needed. It requires multiple steps, and sometimes your cartridge may dry. You have to be very careful. And pressure, we have to be very careful about the pressure. So this is one more technique which we call it as the Spme technique, solvent, solid phase micro extraction technique. And we use headspace Spme technique, especially for volatile samples. And they can be directly coupled with GC technique. So that is one advantage here. This is just a needle kind of thing. Then for polar and for non-volatile substances, we have direct immersion SPME, that is direct immersion solid phase micro extraction. So this is the solid. This is the solid support. This is the stationary phase, or you call it as the solvent phase, which is coated. You can see here, this is this one. This is the solid support. This is the stationary phase, which is coated. What we normally do, this will be like a syringe model. We can see here, this will be like a syringe model. This is a retraction process. So once your plunger under normal conditions, this SPME, that is the needle. The internal surface is the support. The external surface is the sorbent. So the needle will be intact here in this portion. The needle will be intact in this portion. The moment you press the plunger, this sorbent needle will come out. So what we can do, we can directly put it into the sample. So once you put it into the sample, you just press your plunger so that the needle, you can see here, the needle comes out, and just shake for a minute so that the drug gets sorbed onto the sorbent layer. Then you can again pull up the plunger so that the sorbed needle will enter into this portion. You can remove it. Then you can put it into directly for injection, or you can just put it into another solvent, a vial containing another solvent, so that your analyte is dissolved into the solvent, which can further be used for analysis. Suppose if it is a volatile analyte, you can directly inject it into a GC. So when you are directly injecting, what happens? You press the plunger, your sorbent will be exposed, and because of the heat of your injection unit, the analyte will get evaporated and it will enter into the GC for further analysis. This is one. Yeah, so this is how the thermal desorption and on any GC injection port. We are not doing anything. This is H-space SPME. So you put it into the sample, you press the plunger, the analyte gets sorbed onto the needle surface, you retract it, then you directly inject it onto a GC column, injection port. Or else what we do, you just put it into, this is a direct injection, that is some liquid will be here, you inject into that so that it gets dissolved and it gets analyzed on a HPLC. So these are some of the Spme cartridges which are commercially available in the market, like polydimethyl siloxanes, PDMS, and all those things. PDMS with different film thickness and all those things are available. It has got its own advantage. It is a solventless sample preparation procedure. Simple, straightforward procedure, only sorption and desorption. It is compatible with chromatographic analytical systems, and it is an easily automated system. Sampling devices are portable, so they enable their use in the field monitoring, and it facilitates unique investigations like extraction from very small quantity of samples and all those things. Disadvantage, sometimes some matrix will also get absorbed. If those matrices are volatile, they also will get analyzed. This is a stir bar Octave technique. This stir bar Octave technique is a kind of your magnetic stirrer kind of thing. So you may have an iron bar, and onto the surface of the iron bar, the solvent is coated. So what you can do if you have a large quantity of sample and it is not possible for us to pass it through all your Spme cartridges, and under such conditions, you can just put this stir bar thing into your sample. You can apply you can operate it like a magnetic stirrer, so that when your sample is swirling, all your analyte will get absorbed onto your sorbent material. So most commonly used is polydimethyl siloxane, PDMS. It will get absorbed. So equilibration is very important. So you give sufficient time so that all your analyte gets absorbed. Then you remove that stir bar, you dissolve it into another solvent, and then you use it for analysis. Yeah. So these are some of the very basic sample preparation techniques which are used in case of studying environmental samples, as well as in case of studying some of the biological samples. And thank you very much, all of you, for your patient listening.
Thank you, ma'am.
Yeah.
Yeah, here are some questions to you, ma'am.
Yes, ma'am.
Dear participants, if you have any questions, please give in chat box. And here is one question from Krishna Deepika. Are sorbent and adsorbent are different? They are both used for material recovery.
They are not much different. We are just using the term sorbent. The reason is because we do not know whether the process is adsorption or the process is absorption. So we are not much aware, no. So that's the reason we use the common term sorbent. It can be any process. The drug may get absorbed or the drug may get absorbed. Anything can take place.
One more question.
Yes, ma'am.
Yeah, ma'am. Thank you. One more question from Sarala. Are masking agent and precipitants are same?
Again, there are two types, ma'am: masking agents and precipitants. Masking agents can convert your analyte into a soluble complex, or it can convert it into a precipitate also.
Yes.
So both the possibility is there with the masking agent. Precipitating agent is an agent like if you are using acetonitrile will completely precipitate the proteins.
Depends upon the activity.
Depends upon, yeah. Depends upon what we are taking.
Yes, ma'am. Yes, ma'am. Yeah. From your slide, I came to know that you have UPLC MSMS in your lab, ma'am.
Ma'am, actually we have we don't have UFLC. We have ultra fast.
Okay.
So UPLC work we have carried out. Means it was an industrial project what we have carried out.
Okay, okay. What I came to know from your presentation is just you are doing almost all the sample preparations with the MS.
Yes, ma'am.
Yeah. That's what you have covered so much of thing, ma'am. Like just only liquid extraction and protein precipitation only we do generally in the outside labs also even.
Yes.
I mean to say generally for HPLC, whereas mass is, yes, ma'am.
Because, ma'am, SP is a little expensive technique. Cartridges are very costly.
Yeah.
So they normally, any laboratory, even we do start with protein precipitation, and not much of LLE we do, because LLE takes a lot of, it tests your patience.
Yes.
Selecting the solvent.
Getting the solvent itself is a difficult
Yeah,
task for us.
yeah. And after that, we need those nitrogen evaporators and all. So we don't have that facility here. We have to go for evaporation on a water bath. So we may lose the we may lose the analyte. Unlike may undergo degradation. So under such conditions, so we don't try LLE much. We directly go for SP.
That's what actually my next question is, that only. Will you go for liquid extraction or protein precipitation? And thank you for the elaborate explanation on sample preparation, wherever, like almost we covered the entire first unit of the second sem actually, M-Form second sem. And not only that, it can be useful, like, so most of the scholars, they are doing outside because of not having proper facilities in normally in academics. So generally they should have a grip on those solvents. Like they need to know about the solvents at least to say their presentation strongly.
Yes.
That's what I feel. And broad explanation on solvent selection. And you almost covered that Snyder. Apart from that, some textbooks are there for your reference, it seems. We want to know about those references.
There is one book called, I think I have given that there is one sample preparation technique. Mitra is the author of the book.
Okay.
So yeah, that book is there. We can use that.
We have one more book other than Snyder, Mitra, right?
Yeah, so it's a open access book only, ma'am. It's available online, which I have downloaded some 1 or 2 years back. A few of that I have used.
Yeah, ma'am. You tried a lot to finish your topic very fast with me. Right? Last advantages and disadvantages. So many things you have covered in the PowerPoint presentation. Thank you so much for your broad explanation on these topics, ma'am. Whatever we have selected, we justified. I feel so.
Thank you, ma'am. Thank you so much.
And principal, ma'am, she has gone for interviews, ma'am, to attend, to conduct the interviews in the academy. And one more question from our student: temperature and pH to maintain during protein precipitate. Whether to maintain temperature or pH? I mean,
pH is
pH precipitation technique.
Yes, ma'am. Temperature normally we don't use, because we all are aware. Suppose if you take a plasma sample and you heat it, proteins will undergo precipitation. But temperature and pH selection is very, very important. It may lead to degradation of our analyte. One. Second thing, temperature, if we put, the analyte may undergo degradation. We do not know the nature of the analyte, then we have to be very careful with the temperature. pH, nature of the analyte is important. Suppose if you are using pH for protein, you can use acidic acids or bases when we are using. If the drug is ionizable in nature, we will lose the analyte. So it is we normally prefer acetonitrile or methanol, much easier precipitating agents. Yeah, so that the analyte will stay in a non-ionizable form and can easily get extracted in the organic solvent.
Yes.
And if the recovery is not much high, then only we go for these pH and all, changing the pH.
Thank you, ma'am. Thank you for the nice explanation of all the questions. And thank you, Sabina, for asking this question. And thank you so much, ma'am. Ma'am, I think you got struck. Anyway, thanks a lot, ma'am. Now I think,
Power fluctuation was there.
I got it. Thank you very much, ma'am.
Thank you, ma'am. Thank you so much.
Okay, if we have any questions in future, probably we'll approach you along with our students.
No problem. No problem, ma'am.
Moreover, we want to go with these type of techniques, and drugs are not available. Actually, the problem is that one also, like small amount of samples also. If we are getting also, we may not be knowing whether it is pure or not. That is one more target we have in analysis.
Yeah, that's why we don't do all trial and errors. We try to understand the sample nature, and then we try to fix with 1 or 2, because few of my students have purchased 10 mg of drug is around 1 lakh, 1.5 lakhs. So trying one other technique, we cannot waste it, no?
Yeah. And by taking the simple drugs and want to go for publication means, again, it's a difficult task. This is what the
Not
problem
possible,
we have. Yeah. Anyway, while by collaborating with other subjects, we are thinking so, and we do the same. Thank you, ma'am.
Thank you, ma'am. Thank you so much for providing me this opportunity. And thank you all, dear participants, for your patient listening for the past 1.5 hours. I hope it is more
No,
than
ma'am.
1.5
Actually,
hours.
yesterday we got to know about LCMS, instrumentation, and further.
Okay.
Whereas today we got clean explanation on sample preparation, completely whatever we have in the academic session.
Yes,
right,
ma'am.
So in an examination point of view also you have covered, I think.
Yeah, I have covered. Yes, it is. Yeah, as you said, it is there in first term form, second semester, first chapter, I think.
Biological analytical techniques. In that subject,
Yeah.
we have first unit. And
Yeah.
I thought it will be helpful for research scholars in
Yeah, it will
knowing
be
the theoretical part about sample preparation. You almost covered micro extractions also.
Yeah. Feel
very great, and it's privileged to us. Thank you so much.
Thank you, ma'am. Thank you so much.
Yeah, you please, you can
leave. I can leave the meeting, ma'am.
Sure.
Yeah. Thank you all, dear participants. Thank you so much, ma'am. Thank you. Please convey my regards to Sunita, ma'am.
Sure.
Thank you.
And dear participants, you please fill your Google attendance form or feedback form, whatever you have given the WhatsApp group. Thank you so much.
Ma'am, can I end the meeting?
Yeah, sure.
Okay.